I got connected. Can you get it connected? Yeah, please. You go ahead. Then we'll start. Hello.
Yes, sir. Please start the session.
Yeah, Kranti. Yeah, he got connected. Jai Shekhar, you can just start. Okay.
He is connected. Admit. You have to admit.
Dr. S. Jayashekhara, he is going to address welcome note to the participants.
Am I audible?
Yes, sir, we can hear you.
Yeah, distinguished dignitaries, invited guests, participants, ladies and gentlemen, I would like to extend a very warm welcome to every one of you and welcome you to the 3-day open webinar, Plantation Crops, Genomics, and Overview of the Current Research, jointly organized by the Indian Society for Plantation Crops and Bionivid Technology Private Limited, under the overarching umbrella of ICAR, Central Plantation Crops Research Institute. It's a glorious moment to extend my warm wishes on behalf of Indian Society for Plantation Crops, as the year 2021 marks the onset of the golden jubilee year of the society. A memorable travel of 50 years with rock-solid commitment towards the cause of sustainable growth and development of plantation sector of the country. It's a great field that all evolving and vibrant young Bioinvid Technologies, the specialist data analytics organization with key focus on genomics application, is co-hosting the event. I thank you all for being with us today. We are well aware that having assimilated the unprecedented events followed by the pandemic all through the year, and the reverberations are still on, but our togetherness reminds us of one thing, that knowledge has the inherent power to transcend any barrier. With this note, I would like to express my wholehearted welcome to Dr. Anita Karun, Director ICR Central Plantation Crops Research Institute, to this event. Ma'am, you keep on influencing us with effective guidance and excellent leadership. I cordially welcome Dr. H.P. Maheshwarappa, President ISPC, who is leading the Indian Society for Plantation Crops with the sincere and keen concern for the betterment of the plantation sector. Welcome, sir.
Thank you.
Most courteous welcome to Dr. Rohit Shukla, founder Bioinvid, who is partnering with ISPC to co-host the open webinar. Welcome, sir. We are honored to have most respected Dr. P. Ratnam. I'm unable to see whether he is there or not, but he is keen to got registered. So if he's there, warm welcome from our side, sir, on behalf of ISPC. He is the former Director International Coconut Community. We have scheduled 9 technical sessions spanning around 3 days, and have with us eminent and expert resource persons who are sure to enlighten us with their expertise and experience. I welcome all the resource persons to this program, and especially warm welcome to our overseas scholar, Ms. Liu Shuli. She'll be joining with us now. He's head of commercial AP for bringing the expertise to our gathering. We have registered participants across India and overseas. This event would have been impossible without the support of each and everyone present here. It gives me immense warmth and pleasure to grace all your presence. I sincerely welcome you all to the program. It's an opportune moment to refurbish and debate upon the problems of mutual interest with our esteemed resource persons. The seminar schedule brushes upon a spectrum of exciting and benefiting themes, hope so, that discusses the aspects of cutting-edge frontiers of plantation crop genomics in particular. This event would have been impossible without the support of each and everyone. Once again, I am reassuring that I ensure that this seminar will be profitable and fruitful for everyone present over here. Thank you, one and all. With this note, I also would like to call upon Dr. H.P. Maheshwarappa, President of Indian Society for Plantation Crops, to offer the presidential address. Thank you, one and all.
Thank you, Dr. Jayashekhar, for welcoming the distinguished members and guests who have come registered for this open webinar on genomics of plantation crops. Distinguished members of ISPC, invited guests, ladies and gentlemen. Am I audible? Hello, am I audible?
Yes, sir. Very much.
Very much audible.
It is pleasure to start this auspicious occasion of ISPC for completing the Golden Jubilee year. This is a Golden Jubilee year. We have started with this first webinar series, and we'll be conducting more scientific webinars and seminars in the coming days to mark the occasion of the 50 years of completion of formation of the Indian Society of Plantation Crops, CPCRI Kasaragod. As you all know, we are facing the pandemic COVID-19 for the past 1 year. Even with that, I think we have learned a lesson to how we should keep our environment clean and how to protect our environment. It is a big lesson for the human community. And with this, definitely the coming days will be having a very good pleasure, and with the onset of the COVID, this vaccination, I think we need not fear much, and our life as usual will be beginning to start with all the economic and other scientific activities. As you all know, ISPC has been working in improving the plantation crop sector with the scientists across the country and abroad, and has grown to be the premier scientific society in our country. And ISPC professional forum devoted for sustainable growth and development of plantation crop sector, established in 1971 by the founder, director of the CPCRI, the late Dr. Ahmad Bawapa. The society has presently metamorphosed into one of the premier scientific society in India. The activities of our society are publishing the journal, one of the eminent best journal in the horticulture society, the Journal of Plantation Crops, and holding national and international symposia. We have conducted many international symposia on plantation crops, and in the series, the biennially, we are conducting the plantation crop symposium that is called as a Placrosim. So far, we have conducted 23 Placrosims, and 24th is on the online. And with the publication of the Journal of Plantation Crops, which is a peer-reviewed and having high NOS impact factor, and it is covering all the crops like our plantation crops, coconut, arecanut, oil palm, spices, and medicinal aromatic plants. All the crops will be covering in this Journal of Plantation Crops. ISPC in collaboration with research and development institutions in the field of plantation crops organizes this, as I told you, Placrosim, and the next Placrosim will be hosted by our sister organization Spices Board, and the probable dates will be announced in the coming days. And as you all know, the 2021 marks the onset of Golden Jubilee year of our society. We have taken the first initiative to celebrate this milestone achievement in the best possible way by sharing and spreading knowledge through a 3-day open webinar on plantation crop genomics and overview of current research. Friends, as you all know, the research technology advancements have substantially expanded our ability to analyze and understand plant genomes and to reduce the gap existing between the phenotype and genotype. The fast evolving field of genomics allows scientists to analyze thousands of genes in parallel to understand the genetic architecture of plant genomes and also to isolate and genes responsible for the mutations. Furthermore, whole genomes can now be sequenced. As you all know, the whole genome sequencing work has already been done in many annual crops. And in the perennial crops like our coconut and fruit crops, the attempt have been made. And from our CPCRI also, we have done the genome sequencing of our Chowgard green dwarf, and it has been published by my colleagues. And friends, with this brief background, I once again
Hello,
thank you
hello,
all for joining.
Maheshwara sir.
And
Please,
yeah,
hello. Listen to me.
yeah.
Kindly mute the others because of disturbance is much more. Please mute others. Sir, you are not audible. You are not audible, sir.
Okay. Am I audible now?
Yes, sir. Yes, sir.
Yeah,
Yes, sir.
yeah. Yeah, friends, sorry for the interruption. This is all happens because there is some internet problem and a small HF will be there. Then, friends, I hope that the 3-day webinar will open a era for all the young scientists to look into the concept of genome in our plantation crops, and they will be exposed to by our our speakers in all the aspect of genomics in the plantation crops. And I wish all the best and try to interact with our guest speakers and involve in your future research work. Once again, I thank you all. Thank you very much. Thank you. Please. So next, Jai Shekhar.
Yes, sir. Thank you, sir.
Yeah.
Thank you for your speech.
Yeah. Yeah.
So I would like to invite Dr. Anita Karun, Director ICAR CPCRA, to inaugurate the with the speech to inaugurate the webinar.
Madam, Anita Karun, madam. Hello.
Yeah. Yeah. A very good morning to all.
Thank you, ma'am.
Yeah. So ISPC President, Dr. Maheswarappa, distinguished members of ISPC, invitees, Dr. Jayashekhara, Rohit Shukla, and our respected Ratnam sir, ladies and gentlemen. A very good morning to all of you, and also a Happy New Year. The Indian Society for Plantation Crops was established in 1971 with headquarters at CPCRA Kasaragod, with an objective to provide a common platform for scientists, extension personnel, and other stakeholders to discuss and deliberate various issues in plantation crops. Those crops dealt by ICR Institute as well as those under the commodity boards. They contribute to national economy by way of export earnings. These crops occupy less than 2% of the total cultivated area, but they contribute nearly 13% of export earning in all commodities. However, one may observe that over the centuries, supremacy of India in plantation sector has declined, except for coconut and arecanut. India has not figured in the first or the second ranking of other plantation crops. Further, India is more and more depending import of certain plantation crops like cashew, cocoa, and palm oil. The federal governance seems to be not giving enough attention to crop diversification and promotion of commercial cultivation. To some extent, the deficit in investment in plantation sector that led to poor management of estate, including delay in replanting or rejuvenation, is the main cause of poor productivity and decline in quality. The role of ISPC to bring lost glory in plantation sector is therefore become pivotal. With increasing demographic pressure and urbanization, plant genetic resources are eroding at a faster rate all over the world. It has become increasingly urgent to promote the sustainable conservation of these resources, the plantation crops in particular. This can be achieved through the characterization and evaluation of genetic diversity, seed ecology analysis, and adopting complementary conservation strategies. The advances in genomics research find many applications in agriculture. Plant genomics is a growing and evolving field of study with high power computing facility and data management tools. The hitherto unknown genetic codes are unveiling in many crops. At CPCRA, we collaborate with Bionivid Biotechnology Private Limited, Bangalore, to complete the whole genome sequencing in coconut. We are in the process of sequencing the Aracanut genome at present. At the same time, we are yet to achieve capabilities to use the genome level information in breeding and other branches, especially in disease diagnosis. While doing so, what are the cost-effective protocols? How reliable the mined information is to solve field problems? These are some of the questions arises. And there are questions are many like this. This webinar on plantation crop genomics, an overview of current research being organized by ISPC in collaboration with Bionivid technology is expected to focus on the development and application of genomic technologies to plantation crops and bring forward a roadmap. The topic of the webinar, I feel, is the most appropriate one in the auspicious occasion of ISPC's Golden Jubilee celebration. I compliment the organizers of both the host and co-host institutions and also thank all the registered participants. I wish that everybody attending the webinar would be benefited and enlightened. With great pleasure, let me declare that the webinar is inaugurated. Thank you.
Thank you, ma'am.
Thank you, ma'am. Now I would like to invite Mr. Rohit Shukla, Director, Founder, Bionivid, to share a little bit about this webinar.
Yeah, hello. Am I audible?
Yes, sir, we can hear you.
Yeah. So a very good morning to everyone. And thank you to Dr. Jessica, Dr. Maheshwarappa, and Dr. Anita Karun, Director Ma'am, CPCRI. And really thankful for taking your time out and gracing this meeting. I would like to, I want to convey my heartfelt gratitude to everyone for accepting our invitation and presiding over and participating in the webinar. General webinars are organized and conducted for the researchers as well as the audience to interact each other, where the researchers present their work and which the participants, either students or other researchers, they try to gain insight into what are the current technologies or the assays people are using in the recent genomics or proteomics or in their respective field. And for the so for this particular webinar, when we planned, I had a discussion with Dr. M.K. Rajesh. I would like to thank him a lot for also for providing this opportunity to discuss. So we talk, okay, there are many things in the genomics field currently that has come up, either for the plant genomes, so how to do the genome assembly part. There are various technologies that has come up. So that's why we discussed with them, and then we decided to choose various specific topics where people can get insight into. So each of the topic for the discussion has been curated, keeping the best interest of the young researchers in consideration, so that they get access to the recent technologies that is people are using in this field of genomics. So webinar schedule, just to brush upon that, we have a talk today from Dr. S.V. Ramesh, Principal Scientist from ICAR CPCRI, and who will be talking about RNome for the coconut genome, where he is talking about the low efficiency water and the high efficiency water impact of it, and study from the transcriptomic ways of study. Then Dr. Avatsa from our organization, Bionivid, he will be talking about the role of GBS. It's another application in genomics, in shaping the plant genome, genomics studies or the agriculture studies. And third, we will have Dr. Chueli from Dovetail Genomics. She will be talking about the Dovetail Genomics new technology, that is OmniC. She will be talking about that and how to use the OmniC technologies to, now the power has come where people can build up the genome up to the chromosome level or the pseudochromosome level. So that's something, gives the more power for people to understand the molecular biology, and even people can generate more resources for the other co-scientists and the students to get benefited from. So I would not take too much time. I will just try to close here, and I will ask Kranti and to invite Dr. S.V. Ramesh for his talks. So hope Dr. Ramesh is with us.
Yeah,
Thank you all. And yeah.
Thank you, Rohit, sir.
Thank you.
So I would like to invite Dr. S.V. Ramesh, ICAR CPCRA, to speak on the topic, can transcriptomics offer a way forward? So please take over.
Sorry, am I making it audible?
Yes, sir, we can hear you.
Yeah, okay. Shall I share my screen?
Sure, sir.
It says only host can share this screen in this meeting.
We are making you the host, sir.
Yeah, please.
So you can start sharing the screen.
Yeah. Is my screen visible right now?
Yes, sir, we can see.
Yeah, okay. So at the outset, very good morning to all the learned persons, all the scientists who are involved in plantation crop genomics and others. So I'd like to thank ISPC and Bionivid for giving me this opportunity to talk on transcriptomics, that too in a crop like coconut. So this is an exciting time to talk about transcriptomics for a simple reason that these are the technologies that is giving more information and large volume of data that can be used for many crop improvement programs. And I'm very happy to present my talk. Initially, I thought I'll be talking about water use efficiency and how does it molecular responses of coconut cultivars for water use efficiency. That is what I thought. But considering the context and the audience, I thought I'll talk about
So
the whole
voice is
transcriptomics and
breaking.
Sorry. Am I audible?
Yes, we are audible.
Yeah.
Go ahead. Please, others can mute. Others can mute, please. Yeah. Okay.
Yeah. Okay. So I thought better I'll give an overview of transcriptomics work that has been conducted in coconut. So to start with, I'll just give a perspective of coconut, what it is, and what is the status of our genomic resources, and then I'll go into the topic. And coconut needs no introduction, at least in this part of country, in this part of world rather. So you can see that it is generally grown in coastal areas of tropical regions, and something unique or something interesting is happening with respect to coconut during this part of time. That is, like with respect to cocoa, what happened is that it is widely cultivated in Africa and other tropics, but it is widely consumed in North America and Europe. Similarly, similar thing is happening. The nutraceutical properties and other important aspects of coconut is being recognized in wealthy world, that is like North America and Europe. That is why coconut is getting much more importance. And this is in general about coconut, but for a plant biotechnologist or for a plant breeder, if you see, it offers a lot of opportunities because there are a lot of heterogeneity is there. It's a cross-pollinated crop, so heterozygous nature is there. And besides that, it is a long juvenile phase, and we have issues with in vitro multiplication techniques. And so it offers a lot of opportunities and a lot of things for to enter into transcriptomics, not only transcriptomics, for many other work as well. So And this is a phylogenomic based phylogenetic tree that is being placed here. You don't need Einstein to figure out the output or inference from this particular tree. But I have particularly specifically placed this particular tree for a simple reason that once when your genomic resources are relatively poor, you need to fall back on a distant relative or a close relative species. So that is why I placed this particular slide. Here you can see that our closely related species is L. ghanensis, that is oil palm, or Phoenix dactylifer, that is date palm. These two are almost around 46 million years ago that are diversified. Both crops are diversified, and you don't need genomics data set to arrive at this tree. But of course, even phenotypic and other markers have been used to detect the phylogenomics of this particular crop. But what is more important is that once our genomic resources are relatively poor, what happens is that we need to look for transcriptome, proteome, and other genome sequences from the nearby crops or related crops. So that is why this comes into play. And most of our work in the initial time, 5-6 years back, rather, I would say, it all depends on oil palm and date palm. And let us see what is the status of our genomic resources. Exactly 5 years back, 2016, when I entered a NZBA database, just a search word, Cocos nusifera or coconut, it turned out little over 1,000 ESTs, that is transcriptome sequence tags, plus around 4,000 proteins. That's all, nothing more, nothing less, rather, I would say. But what is the status today? If you see, quite a good amount of gene sets, data sets have been added, specifically with respect to proteins, it has been added. And quite interestingly, you can see that 3 genome assemblies are present. In the right-hand side, you can see that it is right now it is available, which was not available 5 years back. And moreover, that SRA, that is small RNA sequence read archives are there. There again, you can get somewhere around 300 sequence archive reads are there, near close to 400 archives are there. And how does it fare with respect to an established crop or a model plant like rice? If you see, rather it's unimaginable or uncomparable, rather I would say incomparable, rather, because you can see that there are 81 assemblies in rice, genome sequence assemblies, and if you see the SRA, it's more than around 90,000 rather, and projects are too much. But is it fair to compare a crop like coconut to rice? Of course, it's not fair. So how about our relative cousin, closely related species, oil palm? If you see, oil palm once again has got around 8 genome assemblies. And SRA also, if you see, it is more than 2,000 SRA projects are there. So it's still, there is a huge gap between coconut and oil palm. That's what I would like to convey. And moreover, I'm sorry, is there any disturbance? It's okay, yeah. So is it right to compare, or is it, sorry, I'm sorry, it went back. Yeah. So only my idea is it's not the right way to compare a crop improvement program merely based on NZBA database. But what I'd like to convey is that what is the status of our genomic resources with respect to a well-established crop or a closely related crop, or what is how much is the investment, quantum of investment, research investment is being made in this kind of sciences. That's what I'd like to convey. Then, as I said, when we are talking about genome sequences, right now we have got 3 very good genome sequences that is publicly available. And you can see that the first genome sequence of crop, that is coconut, was made available in the year 2017. And the genotype that was sequenced is Hainan Chol, that is a tall cultivar from China. And since that was the first genome, it was a draft genome assembly. And their emphasis was on studying the phylogeny of anti-transporter genes specifically. Why? The reason is that since it is grown in saline environment and coastal environment, they wanted to see the plasma membrane transporters and how does it fare when compared to other related crops. That was their emphasis. And next came Filipino group. They came out with the Katigan green dwarf, that is a dwarf genome. So once the tall genome was available and the dwarf genome was available, naturally they would go for sequence variations. Comparison of these comparative genomics was done, and sequence variations was studied between both these genotypes, and they came out with good number of SNPs and SSRs between these two. There was a very good quantum of data that was added to it. And while all this was happening, CPCRI was also into foray, and we sequenced Chaugad green dwarf, that is a dwarf cultivar, and that was published in the last year, 2020. And here again, it is not just nuclear genome. We went ahead with the mitochondrial as well as chloroplast genome of this particular cultivar. Otherwise, chloroplast and mitochondrial genomes of coconut was already available. And then our emphasis was on disease-resistant gene, because we know that this particular genotype, Chaugad green dwarf, is relatively resistant to an important debilitating disease in coconut, that is root wilt disease. So the 3 different genomes, 3 different emphasis, but I would say that it's a very good genomic resource to add for a crop like coconut. And coming to the BUSCA analysis, this is, you can see here that this is a simple, not a simple tool. This is a tool to assess the completeness of the genome assembly, or for that matter, gene sets or transcriptome. That is, how much we have really covered the genome based on the sequencing efforts. You can see that all the 3 genomes of coconut is available, and it has been compared with African oil palm, American oil palm, and date palm also. And you can see the last column, there are some missing BUSCOs. That is, what we expect is that, based on the evolutionary analysis, we expect some universal, like single copy orthologous genes to be present in the genome, but that is missing in our genome. So there is a gap. What I would like to convey is that there is a gap that still we need to go for resequencing, and we need to further fill the gaps. That's what I would like to convey here. And what does a transcriptome constitute? Now I'm getting into the topic which I would like to talk, or I would like to speak about. And that is, generally, whenever any other scientist other than a biotechnologist or a general, if you talk about a transcriptome, the only thing it comes to their mind is the central dogma of life. Then mRNA, they talk about mRNA, and it is just 2 to 3% of the whole RNA, because there is a whole lot of different world called non-coding RNAs, which doesn't code for protein, but they are the major gene regulators, and they play a great role in gene regulation. That too, not just at cell level, it plays at phenotypic level as well. And there are quite a good amount of data or the information that has been generated specifically with respect to non-coding RNAs. Initially, it was like small RNAs, small interfering RNAs, microRNAs. Then came repeat associated RNA, NATC RNAs. N number of RNAs are there, and even long non-coding RNAs that in tree crops that has got a greater role to play. So when we say transcriptome, it is not just mRNA. It involves a whole lot of different world. People thought that this non-coding genome, that is only few portion of genome codes for a protein, so rest of the genome is junk DNA. It's not junk DNA. Those are the non-coding part that plays a greater role in ultimate final phenotypic expression. And next, how to study a transcriptome. There are quite a good amount of techniques to study the transcriptome. Till some year ago, we used to study using low throughput techniques, even though we used to do it real-time PCR, RT-PCR, and northern blots also we do. And there are some medium throughput techniques like EST-based, like express sequence tags and cDNA clones are available. But when this high throughput techniques came, including microarray and next generation sequencing techniques, it has changed the game, rather I would say, because in a present context, you would get a very good transcriptome sequence data, and with a good amount of bioinformatic analysis also. And right-hand side, given the overall view of how does the transcriptome sequence works, because there are a number of publications are available and number of tutorials are available in the online only. I'm not getting into the details. What I'd like to convey is that we should first fix a biological question, what we are going to study, then go for the right tissue to extract the RNA, extract it. And if you have a genome, right now, once your sequence data is available, then map it to your reference genome. If you don't have a reference genome, go for de novo assembly. Then you look for your quantification of transcripts, and in the meanwhile, you need to annotate it as well, and come out with the final inference. This is an overall idea. And in a crop like coconut, what happens is that, as I said, we are at infancy in a relatively when compared to oil palm or for that matter any other model crop. So here you have a genome as well. And many a times we need to go for de novo assembly also to look for novel transcripts. And there is another way of doing it. There is a hybrid assembly that has been followed for our this also CGD genome work also, where what happens is that it is like the sequences assembled using based on draft genome assembly as well as de novo, and both are combined using bioinformatic tools, so that can be used for quantification, transcript quantification, and further annotation can be done. So this is generally followed for a crop like non-model plants of something like our crop. So the question is, why transcriptome sequencing? So this, I would say, like degrees of per base sequencing cost. This particular aspect has been done to death. We know that it defies Moore's law, and n number of slides are there, n number of people have given talk on this particular aspect, because the cost, I would say, if you have something around 25 to 30,000 today, you'll end up with a very good transcriptome data, which was not possible 10, 15 years back. When Craig Venter decided to sequence his own genome, it was so costly. So it is very much incomparable when you see 10, 15 years back and what is happening right now. So it's relatively cheap, and it is very cheap rather, I would say. And of course, development in computational biology, as I said, there are different ways to do it, including hybrid assembly and other way of doing this. And there are some advantages over conventional and even modern techniques also. That RAD tags like techniques are there, like restriction site associated DNA tags and genotypic sequencing, because what we deal here is functional genetic element. Because the mRNA is a functional genetic element, DNA is just a repository of information. I would like to have an analogy with a library. A library full of books is nothing but a DNA, but which one to read and which one to infer, that is what mRNA makes. So what we deal here is a functional element, functional genomic element. So it makes a lot of sense when to study a gene expression patterns and to come out with a useful inference. And besides that, we can come out with potential genic markers, not genomic markers, genetic markers. I'm talking about genic markers, that is from functional markers. It could be SSRs, since a number of genome sequences are available. We can go for SNPs also. And another is looking for allele mining and allele frequencies. That is possible using transcriptome sequence alone. So all this comes in a relatively less cost compared to your whole genome sequencing, or for that matter, any other reason, GBS or RAD sequencing techniques. And coconut, yes. What is happening in coconut is we are good at that first point, that is basic research, phenotyping for drought, or for that matter, any disease resistance, and identification of biochemical markers and molecular markers, and field evaluation. That is all very good. But when it comes to toolbox, breeders or biotechnology toolbox, we have got array of techniques to use, but our studies are very much limited to SSRs, to some extent to SNPs, only that. So we rarely use genomoid studies. That is, in a way, I would say it's relatively less. So association mapping studies is just for one publication. Not much has been done, but we are into it. And even if you go for linkage mapping analysis also, it's very less rather, I would say. Only handful of publications are there, or maybe 7-8 publications are there. And mostly comparative genomics we do. And coming to multi-omics, we generally go for RNA sequence analysis, and we need to go for metabolomics as well as ketelomics also. And we are into RNA and gene knockout studies. This is the status actually. So once these are clear, naturally we will end up with an improved coconut cultivar. That's what I like to convey here. And now entering into transcriptome studies in coconut nusifera. What happens is, I'm not going to completely review all the studies that is being carried out, but what I feel is that there are some handful of very good studies, rather seminal works, that I like to present. So the very first transcriptome study was conducted in the year 2013, published in the year 2013. So here you can see that their idea was there's a de novo assembly naturally, because by then genome sequence was not available. So their idea was to see leaf and fruit tissue, what is the expression pattern, or what does it happen to the what is the molecular response for that. So they took Hainan-Tal cultivar, and you can see that in the down, they have gone with Elias guinensis, that is oil palm based cDNA sequences and oil palm based eugenic, because by then we do not have much of the data. So they use the data set of oil palm as well as protein data sets of oil palm to annotate and use it for this study. So this ultimately the outcome or inference of this study is that particular gene or particular enzyme has been pointed out that is involved in accumulation of medium chain fatty acids. And it was known already, but only thing is that we get to know the actual molecular fingerprint for this particular enzyme or particular this one. So that is where this study stands out. And we all know the importance of lauric acid, or for that matter any other medium chain fatty acids. Next is another important work, that is de novo transcriptome once again, here what they did is that coconut leaves and seeds were done. But here the emphasis was on RNA-directed DNA methylation. So what happens is that it is not that what is expressed that is important. Sometimes what is being silenced is also more important that in a crop like coconut, where we don't have much information. So in that way, I feel this is a very good work. Rather, it was published in the year 2014. You can see that in the down, that is the green colored ones are from the palms. So that means whatever the genes that have been annotated in embryo, endosperm, and leaf, sizable portion has come from palm-based palm-based analysis, and most of the other things have come from other crops, related crops, be it vitis, populus, not necessarily related crops, other crops rather, I would say. So that is how it progressed. Initially, we went for the related crop, then we used other crops for annotation of it. So the ultimate outcome of this study is that here they were able to identify small RNAs that are involved in silencing of genome, so that methylation is happening, so that a sizable portion of genome is getting silenced. That's what their inference is. And this is one important study, rather, I would say, because this is akin to one of the debilitating disease what we have in root wilt. Similar to that, there is one more disease called coconut yellow leaf decline. So here again, what they've done is they've taken the healthy as well as diseased genotype, and they sequence the complete transcriptome sequence of both the cultivars have been done. And their inference is that they were able to identify it is the upregulation of gibberellin to oxidase that is leading to, because once it is upregulated, it causes oxidation of gibberellic acid, so that reduced growth is there and necrosis is there. And besides this, anti-transporter, ABC that transporters are being upregulated. So that is giving energy to the phytoplasma. So that way, this particular study is important. And here again, one more seminal work, rather, I would say, because in coconut, what happens is in vitro cultivation or in vitro propagation techniques are still at infancy only, although we have made a large headway, but still we wanted to see why embryogenic calli is embryogenic and what is the molecular deal or molecular role of this embryogenic calli compared with the non-embryogenic calli. So here we used Illumina sequencing to identify what is happening exactly with this one embryogenic calli, and we were able to identify a number of transcripts that are involved in this process. And it is not that we have gone further also. So we have studied the small RNA also of this particular biological question. Here we identified a lot of miRNAs that are involved in regulating these transcripts. Now further we would like to go for silencing and validating these miRNAs. That project we are yet to take up, and we have taken up that. Like that has been sanctioned by DST, so we are taking it up that. So we want to see that what is exactly that is happening in embryogenic calli compared with like zygotic embryos as well as somatic embryos, so that we will come out with markers that can be used for improving our. I'm really sorry, I didn't move my slides. Okay, yeah. So so that we will end up with very good data. I'll skip this actually. And here is one more study. This, I would say, it's a very good study in terms of how transcriptome has been used here actually. So what happens is there are two different cultivars. One is aromatic coconut, another is non-aromatic coconut. Aroma, by that we refer to 2AP. That is 2AP is a compound, biochemical compound that gives that aroma, which is generally found in basmati rice or jasmine rice. So what happens is that authors would like to see like how does this aroma happens, or what is the difference, molecular difference between these two cultivars, one which gives aroma and one which doesn't give aroma. So they use the transcriptome sequencing database of coconut, this particular two cultivars, aromatic and non-aromatic, they have taken. And when they analyze the sequences, they were able to identify aldehyde dehydrogenases. There is one nucleotide change which leads to change from alanine to proline, so that the entire protein sequence changes. Therefore, that particular protein becomes non-functional, so that this 2AP is not processed further, so that gets accumulated in coconut water. That is our coconut water. So that gives an aroma. So what is more significant is that had transcriptome sequencing or NGS technology been not there, authors would have used a low throughput technique, like studying a handful of genes at a time. And it would have taken more years to come out with why this is happening. But since transcriptome sequencing is available or this technique is available, they were able to identify, pinpoint what is happening exactly. So that way, this is a very good study and a very seminal study, rather, I would say. No wonder this is published in Plant Science, and I enjoy reading this paper. It is not just data what they are very clear in what they were looking for. That's what I repeatedly say, that what question you would like to answer, that is more important, and your basic biochemistry and basic phenotypic and basic, that is more important to know where to look for and what to look for. So exactly they know like this, what is the aromatic pathway and how does it work. They were looking for orthologous genes of rice. You won't believe me, rice, and they were able to find out a gene in coconut. And probably this is the first time they were able to find an aroma conferring gene in a tree crop. So that way, this study is a seminal, I would say. And one more study, this once again is from our own institute. Here, as I said, that CGD, that the one which we have got a whole genome sequence, this was used for transcriptome sequencing. The very reason is that this particular cultivar is resistant to root wilt diseases. So that was studied, and once again the emphasis was on looking for the R genes, that is resistant conferring genes. So we were able to identify NBSLR genes, list of n number of NBSLR genes we were able to identify using comparative genomics approach as well as using transcriptome approach. So that is one more study, good study in coconut. And coming to our work, we are involved in studying the molecular response of coconut seedlings when it is subjected to water deficit stress, because we know that in the future climatic scenario, we expect coconut to face a problem, water problem. So we would like to develop a genomic resource or molecular markers linked to water deficit stress or tolerance rather. So here what we did is that we used two genotypes, Kalpashri and Kalpataru. Out of that, Kalpataru is relatively water deficit stress tolerant because it has got a good water use efficiency compared to Kalpashri. That is nothing but, Kalpashri is nothing but our Chowgard green dove, which is resistant to disease, but when it comes to water stress, it is not that good. So we used these two genotypes and we went for biochemical as well as RNA characterization. So you can see that there is a huge difference in epicuticular wax content and response in terms of water use efficiency, and of course enzymatic antioxidants are there. There is a huge difference between a stressed as well as control plants. So this was used for transcriptome sequencing. So what we get is that we face problem in annotation, of course. So because as I said, annotation is still a problem in coconut. So we are able to generate this many million base pair reads, and we are analyzing it for development of molecular markers. So we are able to differentiate, see why a Kalpashri behaves the way it does and why Kalpataru behaves the way it does when stress is there. So we're able to identify the differential gene expression patterns, and also we're able to pinpoint particular genes that are involved in these kind of work. So this kind of resistance. So we were able to see aquaporin, PIP1, and ERP. These are the transcription factors and plasma membrane transporters that gives edge to Kalpataru compared to Kalpashri. So, and besides that, we are able to identify genic markers and long non-coding RNAs also, SSRs, and long non-coding RNAs are relatively new for coconut, and most of the tree crops, there has been very well less explored rather. So we wanted to study that as well. We were able to identify some of the non-coding RNAs from the transcriptome sequence data also. We are validating it for their functional efficiency. And so this is overall what is happening in coconut with respect to transcriptome. But there are some flip side to transcriptome studies also. Rather, I won't say it as a flip side. The very question comes is, the biologically important protein or metabolome is not being captured in this. Yes, of course, I understand that. But that is why nowadays, generally people go for
I think Dr. Ramesh is facing some network issues. We'll just get in touch with him. Kindly wait for a few minutes. Respected participants, I would request all of you to utilize this time to drop in your questions. Our moderators will be keeping a track of all the questions that you're dropping in the chat section. So I would request all of you to come up with questions related to Dr. Ramesh's talk. We'll be more than happy to put them in front of Dr. Ramesh. Thank you.
Dr. Rohit, am I audible now?
Yes, sir, you are audible. We can hear you now.
Should I was it like, should I go from here or like?
Yeah, sir, you
I'll
can
share
start
the screen.
screen
Yeah,
sharing.
yeah.
And
I'll give that.
I
I'll
think
give screen share.
the last slide that was on display, you can start from there, sir.
Yeah, exactly. Okay.
Okay.
Yeah.
Thank you so much, sir.
Yeah. Thank you. I hope I'm audible. I'm sorry. I'm really sorry for that. I could not guess that. Yeah. So what is the way forward in Coconut and Transcriptomics? You can see that. So we can use the data for construction of high quality genomic maps. As I said, our linkage maps are very less and we don't have much information in that. So we can go for genomic as well as linkage map studies also, and for further refining the genome assemblies. And besides that, we can look for allele specific expression and post-transcriptional processes, that is I would like to say RNA editing, that has not been studied very well in Coconut. That is one area of research where we can go for it. Besides that, it is not just geo terms, that is gene ontology terms. We should go further for gene interaction information also, so that a holistic information is obtained. And we are aiming for a targeted approach. Like we don't want to take too many stresses or too many biological questions. Couple of questions we would like to take, and we would like to answer that completely. So that is what we are planning for in Coconut, specifically like one abiotic stress and one biotic stress. That's what we are aiming right now. And besides that, methylome and non-coding RNAs of Coconut have been completely, rather, I would say, barring couple of publications, it is rather underexplored. There is a huge information or huge potential is there to use these techniques for these kind of studies. So this is the way forward. And our research group is very big. Of course, I mentioned only a couple of few peoples only. And acknowledgement for our work, naturally, it is funded by CPCRA in-house project. So thank you. I'd be glad to have questions. Yeah.
Thanks a lot, Dr. Ramesh, for this insightful talk on coconut transcriptomics. I'm sure the participants would have benefited a lot from your thought-provoking and insightful detail that you have covered in the presentation. Also, I would like to now throw the platform open for all the participants to drop in their questions. There are a few which we have received in the chat. I would take them up first. In the meanwhile, I would request other participants to kindly keep on dropping their queries in the chat so that we can take them up and bring them to the notice of the speaker. So, sir, we have received a question from Dr. Deepthi, I believe. She's asking how to link metabolomics and transcriptomics.
Should I answer this?
Yes, yes, sir. Yes, sir. That's the first question.
Okay. It's a very good question. Thing is that, as I said, it depends on your biological question. Not always your metabolome is required, actually. So if you are looking for particular this one, for example, a disease resistant, naturally you can go for metabolomics as well. So it is not required in all the studies. For example, in embryogenic ALS and those things, generally we don't go for it, because there it is an undifferentiated mass of cells where we expect only gene expression to play a role rather than a metabolome. Well, it is cell metabolome rather. I'm not talking about the externally what we are giving metabolites. So it depends on your biological question, and how to link is, it depends, as I said, it depends on your question, what you would like to answer. So for example, if you are looking for a drought tolerance, we expect certain secondary metabolites to express. So we can look for whether there is a correlation, whether there is no correlation or what. That way we can look for. That is the way we do for drought tolerance. Yeah, I hope I answered your question.
Yeah, I hope Dr. Deepti, your question was answered. Okay, in the meanwhile, we'll take another question, sir.
Yeah, please.
This is from Dr. Kalyan Babu.
Yeah.
IIOPR.
Yes, sir.
He's asking, is there any report on practical application of transcriptomics validation in coconut?
Practical application, of course, there are there. As I said, let me quote a couple of things. Number one is we have got our own transcriptome sequence data that is for embryogenesis, that is zygotic embryos as well as somatic embryogenesis, that we are making use of. Practical application means we are trying to validate it using RNA knockouts that we are taking it up. And besides that, I'd like to say one more study, that is that aromatic coconut and non-aromatic coconut, they were bang on. They were able to exactly pinpoint what is happening, why a particular cultivar is aromatic or particular coconut water is aromatic, why not. So they were able to identify, rather I would say, gene responsible for that particular trait. Although it's a qualitative trait, but they were able to identify. I don't say that just because of transcriptome only they did that, but what I would say, had there been no transcriptome sequencing studies or no NGS based technology at all, it would have taken further more years to study one by one. So in that sense, I would say that that is a very good validation or very good publication, rather I would say. Yeah. Dr. Kalyan Babu, I hope I answered your question.
I hope, Dr. Kalyan Babu, your question has been answered. We have one more question from Dr. Sandeep Shale.
Yeah. Yeah.
He's from, I think, CPCRI only. Yes.
Yeah.
Dr. Sandeep is asking, he has a question about how to prepare or where can we get the list of sequences of transcripts on reference sequence of coconut for mapping with experimental RNA-seq data for the purpose of identification of differential genes.
Sandeep, since we have a genome, we need to go for genome-based like mapping only. Rather, it is not that you cannot go for de novo assembly, but still, since genome is available, genome sequences available already. So that yes, you can see all the 3 publications. You can see that they are given the sequence coordinates and details are available. That you need to take and you need to go for mapping rather. That is the only way out. And other way out is that you can go for de novo also. It is not that you can look for novel transcripts also. That is also possible. And that is the only resource it is available. That is, genome sequence is the only resource. With reference to that, you need to map your sequences, your output sequences. Yeah.
I hope, sir, Dr. Sandeep, your question has been answered. Sir, as you might know, this stream is also being live. This session is also being live streamed on YouTube. So
Okay.
we have a few questions coming in from YouTube as well. So the first question is from Dr. Madhumita. She's asking, first of all, she is thanking you for providing a wonderful insight to gene expression profile in coconut. She's also asking you about how many biological replicates should we need or do we need to maintain in such an experiment?
Well, it is a very good question. Rather than having a technical replicates, it is better to have biological replicates, at least not less than 3. That I would say that, because of course, more the biological replicates, more it is good, but at least not less than 3. If you have fund constraints, at least go for 2. But I would suggest to go not less than 3 biological replicates. That would suffice you, because nowadays, not necessarily journals, even if you want to make something useful or useful inferences, at least you need more the replicates. It is good. So not less than, I would suggest at least 3. If nothing works, at least 2 rather. Yeah.
Okay. I hope it answers your query, Dr. Madhumita. We have one more question coming in from YouTube. Dr. Amruta Lakshmi, she's asking how transcriptome analysis data can be used for marker-assisted selection for disease-resistant palm.
Amruta, it's a very good question. Rather, what happens is that when we look for genomic markers, generally we have genomic markers. We use SSRs and other also, for that matter, SNPs also we use. So what happens is that when we have a contrasting two genotypes, or for comparative transcriptomics, if we do like healthy and diseased forms, here what we get is that even from the transcriptome sequence also we can identify SSRs, that is genic SSRs, or genic SNPs also are possible. So that way what happens is that whatever the markers we identify based on transcriptome sequence, they are based on functional elements. So naturally they have a greater credence compared to genomic markers. So that way we can make use of that. That's what we are using it for Chaugard-Greentroff based root will disease resistance program. That's what we are making use of. I hope I answered your question.
Yeah, I hope your question was answered, Dr. Amruta. I think. Any more question, guys? Yeah. So I think that is pretty much it. Thank you so much, sir. Thank you for taking out the time, and thank you for providing us with such an insightful vision into coconut transcriptomics. Thank you so much, sir.
Thank you. Thank you so much for the opportunity. Yeah.
I would request Ms. Kranti to take over now.
Thank you, sir, for the wonderful talk. And now we have with us Dr. Awadh Shah. He's working with Bionivid, and he's going to deliver a talk on genotyping by sequencing. Dr. Awadh?
Yeah.
Yeah.
Good morning. I'm here.
Yeah.
Are you able to hear me?
Yes, sir.
Yeah. So I shared my screen.
Yes, sir. We can see the screen.
Yeah. Good morning to all of you. I'm Dr. Awadh Shah. I'm working as a head for the Genomics R&D at Bionivid Technology Private Limited. So today I'm going to discuss some points about genotype by sequencing, which can be a perfect tool for the breeding and the seed selection criteria, especially for the plant breeders and the biologists. So to move forward, So basically, what is GBS? It's a very simple, highly multiplex system for constructing libraries for next generation sequencing. Now, it is an advanced technology than the RAD technology, which is developed to reduce sample handling procedures, to reduce the PCR steps and purification steps. The main important thing is like there is no DNA size fragmentation in this. It also has an effective barcoding system to differentiate between different samples. And it also is important for simultaneously marker discovery and genotyping. So Cornell Center was the first, like Ed Buckler is the scientist who came up with the first paper in 2011 for a robust, simple genotyping by sequencing technology for high diversity species. So in this paper, they discussed how GBS technology was better than the available RAD technology, and how they can how they can reduce the complexity of the genomes, genome complexity, how it can be reduced through this GBS technology. Now, before going into the details, I'd like to go through some of the common terms which is been used. Like GBS is also known as genotyping by sequencing. It is specific method out by Ed Buckler's lab at Cornell. Started the buzz around low-cost method for moving genotyping to the high-seq Illumina sequencing. Second was RAD sequencing, was developed by Keygene and Florex. It was used for the development of SNP discoveries, basically for the different plants, and it has clearer and deeper coverage than GBS. It is also used often for ecological studies, phylogenetic, or projects where the SNPs are later developed on an array, chip-based systems. Then the third is the scheme sequencing, which is what's created by Huang et al. In 2009. It was termed later after that, after the publication. It is used when we have very low coverage below 5x depth. So at that time, the scheme sequencing is being used. The fourth one is targeted sequencing or exome sequencing. It is any number of methods which are commercially available, but they are very high priced, this exome sequencing. Now the background of the GBS is GBS in any large genome requires reduction of genome complexity. So to overcome that, there are two methodologies available. One is the target enrichment, which is a PCR based, where we do the long range PCR for specific genes or a genomic subsets. And we use probes. It is an RT-PCR based method. And then we do the sequencing capture approach during hybridization, which is also microarray based. Now, the another method which is now latestly used, it is a restriction enzyme based method, which is technically less challenging, where we use methylation sensitive restriction enzymes to filter out a repetitive genomic fraction. It produces different fractionation in the genomes at random, and it gives a random fragmentation. So there is no site selection, and all the fragments, we can do the selection of those fragments. For example, from 100 base pair to 600 base pair, if we want, we can do the site selection. We can we can do the pooling, and after that we do the sequencing of that. Now, why we are using restriction enzymes and why it has been successful? It has been known that a lot of QTLs are located in non-coding regions. They are also located in not only in exomes, but also on the non-coding regions. So that's why these restriction enzymes are used to map large number of genome-wide markers, which are located on these QTLs. Now, this is the overview of GBS. Like we have restriction sites. Wherever the restriction site is there, we add the sequence tags in that, and we do the sequencing of that. And if you have two samples with different restriction sites, for example, which is shown here, then we can also score the SNPs which are not available on those regions. So the idea of GBS is to focus on the restriction fragments and sequencing of them, and to score the SNPs, presence and absence markers also. So this is the example of RAD versus GBS SNP, a basic workflow, where if we do a RAD sequencing, first of all, we have to digest, then we ligate the primers, then we do the pooling, then random shading is there, size selection is there, then we ligate Y adapters, then we do the PCR of this. Now, if you see in the GBS SNPs, like if it is there, then we do the digest, ligate adapters, we do the pooling, and then after that, we directly do, from pooling, we directly do the PCR. So it means that like if we do the GBS, like the steps of like random shading, size selection, ligation through Y adapters, these things are not needed, which were very tedious and challenging during the RAD sequencing. So when I discuss with many of my colleagues regarding the GBS technology, the basic thing, the first question which I'm being asked always is like how much SNP I will get if I do a GBS. Now the answer for this is like it always depends on the type of the plant. Now the things in which depends is like the genome size of any crop, the expected and expected heterozygosity, which affects the size of the fragment pooled for desired amount of sequence coverage. Also, it depends on the enzyme choice which we do during our sample preparations, and the multiplex level what we want to do. It also depends on the genome extensive diversity and how well the samples are captured at that diversity. If now GBS has been always used in both the ways, where we have a reference genome and where we don't have a reference genome also, then that in those cases also we can use the GBS technology. But if we have a reference genome, there are always chance that we will get 3 to 4 times more SNPs by aligning it to the reference genome. The GBS methodology is very simple. Like we have to first finalize which are the restriction enzymes which we want to use. There are a lot of examples available. Nowadays, a lot of case studies has also been done, like which of the enzymes use the higher number, which of the enzymes has higher repeated restriction sites, and which can use the higher copy number of the fragments in that. For example, like I have shown some examples for plants and animals both. Now, this restriction enzyme is used for used to remove the complexity of the genome. So first of all, we do is digest the DNA with restriction enzyme. Then we ligate the adapters. Then we pool and amplify samples. And then after that, we do the sequencing. Now, sequencing is been done normally at around 10x or 20x, 20x depth in Illumina sequencing. And after that, we do the analysis of them. So this is the slide for adapter design, which we use for the barcode adapters. And there are two types of adapters, like one is the barcode adapter, second one is the common adapter. So during the barcode adapters, it's been wherever the restriction DNA insert is there, we add on one side where there will be a barcode adapter, and on the second side, it will be the common adapter in there. So barcode adapter always binds to the 3-days strand of the 3-day strand. So what is the mechanism? The mechanism for adapter ligation is that, like barcode adapter, if there is a parent one, we as a barcode which is of around 4 to 8 base, it shows the sticky end, and on this and on the sequencing primer, there will be on the second primer we'll be adding the common adapter in that. So after doing this barcode, we do the plate DNA and adapter pair. Then we digest the DNA with any of the restriction enzyme. We ligate the adapters. We pool the DNAs and we do the cleanup, PCR cleanup. Then we perform the PCR. We clean up the PCR and evaluate the fragment size for this. Now, GBS has also been optimized for many of the species. Like basically, the idea is like if you are working on novel species, then you need to first identify which is the right restriction enzyme, which can help you to get the maximum number of fragments in that. So, first it will be the identification and testing of potential suitable restriction enzymes. After that, we'll be optimizing reaction conditions, which can give the higher number of fragments in it. Then we do the adapter titration, digest DNA, and ligate variable amounts of adapters, PCR amplification, and evaluate libraries for the fragment sizes. Now, GBS captures barcode and the sequence, and they are doing. The sequence are being done in a single read only. We don't have a in Illumina. If we are doing whole genome sequencing, we have there are different reads available. There will be only one barcode. There will be one restriction enzyme, and there will be a single read there. Also, there are variable lengths of GBS barcodes. To have a good design in modulating the restriction enzyme cusp positions with variable length barcodes produce even the distribution barcodes for that. So if you see, like in GBS, we have different insert DNA with different restriction enzyme, and according to that, the barcodes are being designed, which are of the different 4 to 8 base pair fragment, and we add them on the GBS. Now, to design a barcode, what are the things which we need to take care? Like barcodes are a set of enzyme specific. They are a set of enzyme specific. It must not recreate the enzyme restriction size. Must have complementary overhangs. They are the sets must be of variable length, like from 4 to 8 or 12 nucleotides. Now the bases must be well balanced at each position. Must have different enough from each other to avoid confusion if there is a sequencing error. It means like in between two barcodes, there is at least 3 base pair difference should be there, and must not nest with the other barcodes. It should not mix up with the other barcodes. Now the GBS does not use the Y adapters, which are normally used in the Illumina sequencing, because the size of the fragments are different. So that's why the barcodes which are being prepared for GBS are a different one. Now the same ended fragments, these denatured libraries, their flow cell with they are bound with the oligos. Then there is bridge amplification done. Then cluster formation and linearization. and we get the cleavage from the surface. Now, what are the advantages? Like to have a GBS, like it gives the sequence predetermined area of genetic variation over many samples. It provides low cost per sample for certain applications. It reduces uncertainty basis compared to an array. It identifies variants other than SNPs, especially like insertion, deletion, and microsatellite markers. It enables comparatively analysis across samples in the absence of a reference genome. It informs genetic maps. It is used in screening backcrossing lines. Purity testing is also it is also used in constructing haplotype maps, association mapping, genome-wide selection of plant studies. In all these cases, GBS has been widely used. Now, what are the pipelines for GBS? So basically, the pipelines are being defined in 3 sections. First, we call the discovery pipeline, in which we require a reference genome, and there are multiple steps to process the data into genotype. The second one is the production pipeline. It uses info basically from the discovery pipeline only, and it is faster than the discovery pipeline. And it is only one step from sequence to genome. Then the third one is UNEK pipeline. It is being used for the species which do not have the reference genome informations. So basically, this is the chart for the pipelines where discovery, like where we get the fast QC file, then we do the text by taxa and text count total. Then we do the topping of them, then SNP column, genotypes, and filtered genotypes. In the production, there is fast QC, then we do the top PM mapping, and then after that, directly the genotypes. The third is like UNEK, which is Universally Network Enabled Analysis Kit Pipeline. It is designed for the species that lacks reference genome. Also, it is if the genome is like diploid or polyploid, in which this pipeline is used. If we want to, for the inbreeders and outbreeders, also this, and also for those which are having limited genetic or genomic resources informations which are not available in those kind of genome, this pipeline is being used. So this first is like we filter by coverage, then we filter by taxa coverage, then we filter by the heterozygosity. Then the next filter is added by local LD linkage distribution. Then we have HapMap, and then we do the GWAS analysis for this. The widely technology which is being used in plant breeding is genome-wide association studies, which is linked with GWAS analysis. So before going too deep into GWAS, like there are 2 kinds of mapping which is being used. One is the linkage map, which is being made, and the second one is the association map. So the linkage map or family mapping, like in which we do the generation of mapping population, it is being used for rails, nils, and by crossing F2 populations. We can create a phenotypic markers, and it's also the phenotypic trait of interest. According to our trait of interest, we can characterize the markers in there. But the resolution limitation is that like it has a resolution power is very low, and it works in small population size. Modest degree of recombination, if the modest degree of recombination is present within the population, then only it will work. Now, linkage mapping, it is limited to sampling only 2 alleles at a given locus in any given biparental population. The second one is the association mapping, which is like it is currently existing natural populations are used versus generating a population via biparental cross. There is no need to develop a mapping population. A potentially large number of allele per locus, like in linkage, like we need to have only 1 allele per locus, at least 2 allele per locus. Here we can have large number of alleles, as suppose as only 2 can be served simultaneously, and resolution can be dramatically increased. Like we can have very good fine mapping. It also reduces time. It is used in considering recombination of history, and it's also used in the evolution studies also. The associated mapping has been a application in multidisciplinary field like genomics, genetics, molecular biology, statistical genetics, and even in bioinformatics. Now, what are the basic resources which we need for Jivas? Like first one is the jump class choice. Then there is second is the trait evolution. The third is the identification of the candidate genes. Fourth is the estimation of the population structure. And fifth is the statistical analysis. The jump class choice, like which is the very much important, like as it encompasses, like if when we are selecting any germplasm for GBS studies, we need to encompass as many as phenotypic variations as possible to represent the complete breeding pool of that crop species. Now, for these kind of studies, people have used earlier studies, if you see, where people has used breeding, like phenotypic variations of different locations from different countries also, when they will to get a bigger phenotypic variation picture. The second is like genotypic or phenotypic surveys can be used to identify genotypically diverse subset of all the available germplasm in order to maximize the range of alleles sampled in the population. In some species, there are core set of germplasms that have already been defined and characterized. That things can also be used in our germplasm choice. The germplasm should have maximum diversity in a gene pool with more extensive recombinant events. Now, the linkage discipline, these are the non-random association between alleles at different loci. The LD extends to much longer distance in self-pollinated crops than in cross-pollinated crops. Genome-wide LD determines the mapping resolution and marker density for a genome scan. So the plant population enrolls for association studies can be classified into 5 points. First is like ideal samples with subtle population structure and family relatedness. Then multifamily samples, sample with population structure. The sample with both population and familial relationships. And the samples with severe population structure and familial relationships. Now, as in India, we have very much local adaptations and selections, and because of our breeding history, in many of the plant species, many populations for association mapping falls under our fourth criteria, which is sample with both population structure and familial relationships. Now, what are the factors which affect this LD? First one is mutation, base for producing polymorphy. Then second one is the recombination. Third is the population size. Fourth is the population mating system, self or across. Fifth one is admixture, introduction of like chromosomes of different ancient trees and allele frequency. Then fifth one is the selections. Now, candidate gene markers, how we select them. Like it can be maybe related or unrelated. Then it can be comparatively genomics and it should be developed from completely genomics and bioinformatics. SNPs are related. SNPs are always the preferred marker system in any candidate gene approach. There also can be promoters, introns, exon, 5'3" UTRs, which are targets for the identifying candidate gene SNPs. Rate of LD decays that denotes the number of SNPs per unit length. It is not essential. It is always not essential to screen all SNPs. SNP that cause phenotypic variation due to the alter in the protein function, like the coding SNP or gene expression, should be our top priority. It will always give us the. It can always be represented as a polymorphic SNP markers. If there are blocks of several SNPs in a significant LD, then there is no need to do all SNPs. And alternatively, select a score of fragmentation of SNP, tag SNP that can capture most of the haplotype blocks. So why we, whenever we are doing this whole genome scanning, like SNP markers, if there is a large number, the resolution is more. Now for SSR marker, which are always in small numbers and the resolution is low for them. Now the third is the phenotypic data. It is also important in the JIVA studies. As it represents a relatively large number of diverse accession, phenotypic data should be collected effectively with effective field design with incomplete block design, appropriate statistical methods, and the consideration of the QTL into environmental interaction should also be there whenever we are doing the phenotypic data. The fourth is the population structure. This is a statistical method, like we do the population structure through structure association, which uses a set of random markers to estimate population structure Q, and then incorporates this estimate into further statistical analysis. The second is the approach is mixed model approach, where the random markers are being used to estimate the population structure and the relative kinematic. And the third is the principal component analysis, which summarizes variations observed across all markers into smaller numbers of underlying component variables. The fifth is the statistical analysis. Like basically nowadays the tools which is being used is a TASL. Like TASL is a trait analysis by association evolution linkage, which on every given germplasm, when we have phenotypic and genotypic, then there is a structure. We can prepare a K matrix. We can get the linkage distribution, marker trait association mapping studies also, and the dendrograms for that. Now, JIVAS is a comprehensive approach to systematically search for the genome for casual genetic variations. So these things can be done with the collaboration with TESL. There are a lot of applications for GBS SNP in crop plants. So the major ones I have listed here, which are like for marker discovery, phylogenetic genetic diversity, for fine mapping, for genomic selection, and the JIVA studies. So there are some examples of JIVAs in plants, like JIVAs directly hits the known Mendelian traits also. This example is from the Romi et al. 2013 of genome biology. Even JIVAs is also considered for one of the more complex traits, directly hits for the flowering time also. So if you see in the first figure where the JIVAs was fit white versus yellow kernel in 1,5 USDA MD in traits, it gives a yellow color line of Y1. So the best hit kernel line lies within Y1 Mendelian trait. The second is like with the 660 KSNP, they have done the studies for the JIVAs, and where they find out the different mapping populations, like the NAM population also, which is a nested associated mapping population. And they also were able to find out the largest NAM QTL of the maize. So thank you all for your time and patience. Hello.
Thank you, Dr. Awadh, for this wonderful talk on GBS and associated approaches. I would request all of you to drop your queries in the chat section so that we can take them up and convey them to Dr. Awadh. In the meanwhile, Dr. Awadh, we have a question from Dr. Sumana Banerjee. They're asking, for vegetatively propagated plants, how JIVAs could be conducted? Any suggestions, please? So, Dr. Awadh, could you have, were you able to hear me?
Yes, I can hear you. Yes. So for vegetative propagating plants, like if
Dr. Awadh,
the population set, Hello, are you able to hear me? Hello,
Okay, yeah. Please carry on. Please carry on. Sorry.
hello, hello.
Dr. Awad, you are audible.
Yeah, for vegetative propagating plants, like GBS can be conducted, we need to find out, like we need to first identify the genome complexity in those kind of vegetative propagating plants. Once we know the genome complexity, like whether it is a diploid or a triploid or a polyploid, then we need to see like the how much genetic variation is available within that crop species. Once we find out these kind of variations, then the third will be to identify the right kind of restriction enzymes which can give us the right size of fragments, for example for 100 to 600 base pair. If we have this kind of information, after that the vegetative in those species, we are able to get the SNP polymorphic SNP marker information in that. Hello.
Yeah, I hope it answers your query, Dr. Sumana. We have another question. This is from Dr. Shreya Gosal. They're asking how are the SNPs of pseudogene, how the SNPs of pseudogenes can be detected through GWAS approach. How such SNPs can be associated to LD? That is her question.
Yeah, I got. Now, like for pseudogenes, now, if we want to associate these pseudogenes on the LD, then we need to first identify the SNP, the QTL of that SNP, and the location of that SNP. If that is been identified, we'll be able to map that on that. Also, to identify pseudogenes, like in the diversity population, we need to have a different diversity population, where because of which we can confirm whether the trait, the gene which is available, it is exactly a pseudo or it is an other gene. So if we have that information, then we will be able to link that in the QTL.
Okay. Any more question? Thank you for your answer. I hope Dr. Shreya, Dr. Awad's answer has satisfied your question. Any more questions, guys? Please feel free to ask. Okay, so in continuation to her previous question, Dr. Shreya is asking, pseudogenes not necessarily produce protein product. So how the SNPs can be selected?
Now, see, to get an SNP, it is not always required that we need to have a protein information. There are we can find SNPs according to the like insertion deletion also. There can be various SNPs which are not linked to the any trait markers. So for that kind of SNPs, if we know like on the location of that SNP on the QTL, then we'll be able to link it to the that marker in the LD.
Thank you, sir.
Okay, sir, we have a question coming in from YouTube chat as well. The question is from Dr. Amruta Lakshmi. She's asking, what will be the success rate of using GWAS for identification of markers linked to resistance for slow progressing diseases?
See, if we have very diverse population, like if I see in a SNPs, if normally when we do genome GWAS studies, then the SNP ranges is from like 20,000 to 40,000 to 50,000 according to the crop variety. Now, if we have these kind of SNPs, if we want to identify a specific trait, then we don't need this much number of SNPs. These SNPs majorly are used for the mapping population and to create a LD map and other maps. If we want to identify a specific genes, we need a set of around 8 to 10 SNP genes which can differentiate between this kind of disease, which can either give a positive result if we run those kind of SNP, and secondly, it can give us a negative result. So it can give a positive and negative response in that. Now, to narrow down from these kind of SNPs from around from thousands to around a subset, we need to have a very good phenotypic characters data with them. Now, if we have a different diverse phenotypic character data, and from that, if we use the genotypic data, we can narrow down these SNPs around a set of around 384 or around 198 and or around 16 or 48 also. To prepare those kind of set, the GWAS is always very much widely used. I'm sure many of the people might be knowing around CASP technology, which is an application part which has been done after this kind of genome-wide association studies. So when we do these kind of studies, GWAS, the practical implication is always to have a marker set, SNP marker set, which can identify either a positive or a negative control in that. So if we have a phenotypic variation in that, we will be able to do that. And the success rate will be it will be always more than 50% in that. Hello.
Yeah, thank you for your detailed explanation, sir. I hope, Dr. Amrita, your question is answered. Any further questions, guys? To all the audience in the Zoom session, as well as those who are attending the session through YouTube. If you have any more queries, you can please drop it in the chat. I think that is pretty much it.
Thank you.
Yeah. So thank you, Dr. Awad, thank you for this detailed explanation on GBS approach and answering all the queries that were put up in the chat. Thank you so much for your time. In case if you have any queries regarding this approach or any other sequencing based approach for that matter, you can contact Dr. Awad directly also. Right, I'll just drop in his mail ID in the chat. Yeah, he has already dropped it. So in case if you have any queries regarding this, please feel free to contact him on his mail ID. Next in line, we have all the way from Singapore, Dr. Chui Lee. She is head of commercial Asia Pacific at Dovetail Genomics, and she would be shedding some light on the Omni-C technology, a revolutionary approach in the field of whole genome sequencing. So I think all of you can see her screen as well. So I would like to invite Dr. Lee to take the center stage and address the gathering. Over to you, Dr. Lee.
Hello.
Yeah, you're audible, ma'am.
Okay, thanks. Thanks for the introductions, and welcome everyone to this webinar. First of all, I'd like to thank the organizer, Indian Society for Plantation Crops, as well as Bionivid, for inviting me to this webinar. And my name is Chui Lee. I'm the head of commercial for Asia Pacific for Dovetail Genomics. In case you have not heard about Dovetail Genomics, Dovetail is a leading company, it's a US biotech company, and we are the leading company in building the reference genome. So today, what I'd like to share with you is the technology that we use, as well as to give you a very high level overview of the steps of building the plant genome. So if you have any questions, feel free to type in the chat. And just now I was listening to the previous talk as well, and the talk was talking about identifying SNPs in DEL, as well as looking for the LD markers, things like that. So the talk that I'm giving basically is the upstream steps of what was mentioning just now. So the building of reference genome is basically the first steps of obtaining the genomic information of a specific plant genome. So with that, let me start with my slide here. So what you can see here, basically these are fruits that we eat, but it looks quite different from what we eat nowadays. So this one is basically wild type strawberries, and this is banana, this is avocado, and this one is corn. And you can see that the fruit, this strawberry is actually very, very small, and for this banana, the seeds are like, there are too many seeds, as well as the meat are not a lot as well. And for this avocado, the seeds are also quite big, and these are like not edible. So what we are having nowadays basically are like this. We have beautiful big very red strawberries, very beautiful bananas, corn, so on and so forth. And you probably understand or know that this is because of the crops improvement over time. And take another example of the crops improvement. This is the improvement of a rice variety in Erie, where what you can see in the field here on this left-hand side, this variety that is drought resistant, and whereas on this the plot here, this is the one that actually not resistant to flood. So again, this is actually one of the example where genomic has been used to improve the crops. And we know that the conventional plant breeding is basically done in this type of setup, where insects are basically released into these compartments, and breeding is let occur within these compartments. And we know that these take time, as well as it's probably not that efficient. I worked with one of the oil palm company in Malaysia previously. I'm not directly working with them, but I work with them in a project. And I learned from them that to get an oil palm from planting all the way to fruiting, it takes 10 years. And if, let's say, you are planting a certain seed that is not producing in the quality that you want, for example, the oil production is not high, or it takes a long time to produce fruits, and it takes a long time to know the outcome of the seed. So this is why genomic has become more and more popular, and genomic has been used to improve the plant quality, or use in the plant breeding program. And for example, that some of the plants that we have worked with, many researchers are listed here. For example, you see onion, coconut, maize, so on and so forth. So again, it shows that it's important to get the genome, the genomic information or reference genome, in order to do the downstream analysis, as well as use that genomic information in plant breeding program. So again, like what I mentioned just now, the first steps of getting the genomic information is basically building the reference genome. And my talk is basically to be focused on this part, building the reference genomes or de novo assembly. And then after the de novo assembly, what can be done? So this is genome scaffolding using Dovetail Genomics technology. And now I will share with you a few case studies. Okay, to orientate everyone with the terms that I'm going to use in my presentation, basically de novo sequencing referring to draft genomes or sequencing a novel genome where there's no reference genome available for alignment or for analysis. And then when I talk about scaffolding or genome scaffolding, basically this is linking the non-contiguous series of genomic sequences. Basically, these are obtained from the draft genomes. So linking these genomic information or genomic sequence into a scaffold, which provide the contiguity, the order, and orientations. And I'll talk more about this in my next few slides. And the steps of building the de novo assembly and genome scaffolding are actually quite simple. And the first step, like what I mentioned, the first step of obtaining the genomic information, if there is no reference genomes available at all, the first step is to do the draft genome or to do the reference genome. So this is by doing the de novo assembly. After de novo assembly, we will then take that draft genome and scaffold that, and there is a need of doing genome scaffolding because like what I mentioned, it provides the contiguity as well as it provides the order and orientation to the genes, and that completes the genome. And nowadays with the publications, I heard that there are many publications that require the genome, the chromosome level draft genome as well. And once you get the complete genome, what you will do is to do the annotation. So annotation is kind of like a check to the quality of the genome. So basically, you would like to get as high percentage of the genes that being annotated in the draft genome that you generated. And once annotation is completed, then that open up a lot more opportunity for downstream analysis, such as identifying SNPs that you can link to the phenotypes, identifying indels or other functional studies. Okay. So coming back to the workflow of building the reference genome, again, de novo assembly, genome scaffolding, annotation, and functional study. So for de novo assembly, this is quite common nowadays where you take the plant material, for example, the leaf, and then extract the DNA. And then you can use second generations or third generation sequencer or sequencing technology to generate the draft genome. So the technologies such as Illumina, Oxford's Nanopore, PacBio, these are the technology that are available for draft genome. Okay, now let's take a look on the workflow of the generating the reference genome. So basically, I mentioned that there are many technologies that are available to generate this reference genome or these draft genomes. So you extract DNA, you run it on sequencer, you get the reads, and after that, you assemble the reads into contigs or even scaffold. So from this sequencing reads, you join them together to become contigs, or from contigs, then you join them to become scaffold. And is this complete? Yes, this is for draft genome. This is for reference genome. So what you get is contigs and scaffold. However, like what I mentioned, for draft genome, this is still lack of the order and orientations, and also it's lack of the contiguity. So when I say lack of contiguity, basically it has a short N50. N50 is a matrix used to assess the quality of the draft genome. And why is that so? This is because there are many challenges to build the reference genomes. For example, especially for plants where it is polyploidy, there is a high GC content, there's also high repeats. Some of the species might be very difficult to get the DNA, to get high molecular weight DNA. So these are some of the challenges that causing this draft genome to be lack of order and orientation as well as contiguity. Of course, it's also limited by the technology. And how to overcome these challenges, how to overcome this lack of contiguity, lack of order and orientations. And this is where the technology, the dovetail technologies comes in. So taking a look on the same workflow, now we have the context, we have the scaffold, and dovetail genomics will come into this part where the technology will scaffold this, or will join this scaffold to a longer scaffold that will increase the contiguity as well as to correct the order and orientation. So this is where dovetail genomics comes in. And and with this whole workflow, starting from second generation or third generation technology, and then with the combining with dovetail genomics technology, that built the chromosome scale genome. Okay, so what I'd like to talk is to share with you is the technology, dovetail genomics technology. It's called proximity ligation technology, and the technology is actually quite simple. I'll walk you through this workflow. So first of all, we take the sample, and in this case, for the case of plant, that will be taking the leaf, for example. So we take the leaf, and then what we will do is we will cross-link the leaf material with a chemical. In this case, we cross-link with formaldehyde. And what we cross-link basically are, we are cross-linking the chromatin within the nucleus. So just imagine that this black line here represent the chromatin, and this blue dots here represent the histone in the cell, right? So when we cross-link, because the chromatin are in a cross-link stage, or sorry, is in this folding stage, or in this chromosome stage. So when we cross-link, these chromatin are actually been fixed together, right? So you see this red line here, this represent the fixation or the fixing. After the cross-link, we will then digest with an enzyme. What I'm showing you here is digestions with resistance enzyme. However, there are also many other variations of this technology where we digest with DNAs as well as MNAs. So let me walk you through with this diagram first. So we digest with resistance enzyme. So what you can see here, now we cut this fragment and we leave with the sticky end. So but because of the cross-link, even though we digest this fragment into pieces, they are still bound in this conformation. They are not floating in the reactions that we are doing in vitro, right? So they are still in these conformations. So after the digestion, we then continue to do the end repair. So in this case, from the sticky end, now it becomes blunt ends. At the same time, we also add this green molecule to the end of each fragment. So what you can see here, for example, this fragment, there's at the end of this fragment, there are these green molecule. So same for every single fragment here. So this green molecule basically is the biotin, where we use that for the selections in our downstream step. Okay, so this is end repair. After the end repair, then this is where the proximity ligations happen. So this ligation step is where proximity ligation happens. So for example, this fragment 1 and 2, it can be quite far apart if you look at in a linear form. So let's come back to this very beginning steps. 1 and 2, if you stretch this chromatin in a linear form, they can be separated by megabases, right? However, in these types of conformations, they can be quite close to each other. And because they're quite close to each other, and they have the chances or the probability of ligating to each other. So this ligation step is actually a random process or a random event. So for fragment that is close to each other, for example, these two fragments, if they are close to each other, there is a chances of ligating to each other. So if you stretch them in a linear form, they can be separated in a mega basis. However, in here, they have the chances to ligating to each other. Okay, so this is proximity ligation, and this is the core technology or core event in proximity ligations. So after this ligation step, we remove the crosslink. Now you see that this red line have been removed and we free all the fragments. So for example, this fragment here is free now. So this is the first fragment, this is the second one, and this is the third one. Okay, so now what the fragments that we are interested are basically fragment 1 and 3. Why? Because there is a ligation in the middle. So these are the fragments that we are interested. And 1 and 2, if you look at this fragment here, the one on the left, this part, as well as the one on the right, this part, again, they are from this part and this part. So I mentioned that in linear form, they are very, very far apart. Same for this one. The one on the left, this one is from this part. And then the one on the right, this part is from this part. So again, they can be separated in mega bases, right? And this one is basically a very short fragment, which is like that, which is maybe here. And we are not interested in these types of fragments. We are interested in fragments that are having the proximity ligations, and we know that they can be quite far apart. Okay, after the cross-link reversal, because this is the whole genome approach, we will then move on to do Illumina library prep. So this is just the normal Illumina library prep where we will take this DNA and then we will do the fragmentation and so on and so forth with the Illumina library prep process. And at the end, what we get is we get the library that is similar to Illumina library. However, we know that the fragment that we are getting is not the typical short fragments. We are getting the long fragments. Then with this Illumina library prep, we move on to do the sequencing 2x150 base pair, and then use the software to do the analysis. So what we are looking here is basically we are trying to scaffold the draft assembly. So let's put that into the whole workflow. Now, this is the draft genome, right? I mentioned in the earlier on that draft genome can be generated using Oxford Nanopore, PacBio, or even Illumina. And then with that sequencing rate, you assemble that to become contig or scaffold. So what you see here are the scaffolds that generated from the first step, which is the draft genome. Now with the proximity ligation reads, so what I call this one are the proximity ligation reads, which is from here. Now we have this one. So with this proximity ligation reads, we know that this portion and this portion are quite far apart, right? So when we map this read, back to the draft genome, what we get is basically, for example, this read, when we map to the draft genome, it mapped to this region. And then this read mapped to this region. Same for maybe for this read, it mapped to this region, and then this read mapped to this one. And then we have many, many reads, because when we are doing whole genomes, when we are doing this sequencing, we are getting like maybe 100 to 300 million read pairs, depending on the genome size. So we get lots or millions of read pairs, and then we map that to the draft genome. So basically we map back to this one. And then with this proximity ligation reads, we are able to then group this scaffold into a longer scaffold. So for example, we are able to say that this one, and this one, they belong to one big scaffold. Maybe from this one, and this one, they belong to another scaffold. So this is the theory of proximity ligation reads, and combining that with the draft genome, we now then able to scaffold that all the way to chromosome level. So it could be that this is the one whole chromosome from the species, as well as we are able to orientate that to provide the gene orientation. So this solves the problems that I mentioned previously, lack of contiguity, as well as lack of order and orientation. So these are a few examples that that's using the technology. So N50, like what I mentioned, is the matrix used to assess the quality of the draft genome. So this is a reptile sample, and N50 is very, very short. It's only 500 kilo base pair. And using the technology, using dovetail technology, we are able to scaffold that all the way to 90 megabases. So from million or maybe from thousands of the scaffold, which is from here, which is from here. So there are many scaffolds here. After we join them together, now you see that these are the super scaffold. So in the example here, each of these could represent one chromosome. So this is the pseudo chromosome level. Again, this is another insect with very, very short N50. And using dovetail technology, we improve that. We improve the contiguity all the way up to 151 megabases. And each of these plots here represent one chromosome. So these are the pseudochromosome chromosome level genome assembly. And this is another example with plants from 500 kilo base pair that has been improved to 295 megabases. And these are the chromosome level assembly. So each of these plots, this is one chromosome. Okay, and we know that it's important to have the contiguous reads because that makes the downstream data analysis easier. For example, for gene annotation, for comparative study, or to identify structural variants, as well as if you have the complete genome, it's also easier to get the SNPs information or in-depth structural variants. Okay. So with that, I would like to share with you a few case studies. The first case study that I'd like to share with you is this lactase assembly. And this is actually quite an old publication that was published in 2017. So basically, we know that lactase is an important vegetable crop species, and it ranked one of the top 10 most valuable crops in the US. And for this genome, is a diplot genome, and the estimated genome size was 2.5 gigabases. And we use the technology to scaffold this draft genome all the way to the chromosome level. The methods of de novo assembly and scaffolding was, because this was an old project, it was using Illumina to assemble the draft genome. And then these are the software. SOAP de novo 2 was used for the de novo assembly analysis. And from there, we then use our technology that we call Chicago, and we scaffold that, and then we also use Hi-C to improve that to the chromosome level assembly. So this table shows the stats of the draft assembly as well as after the genome scaffolding. As you can see here, the N50 increased from like in kilo base pair all the way to megabases. this sentence was extracted from the publication itself. So using the technology, it increased that contiguity as well as it improves that order and orientations of the scaffold. And using the Hi-C, we will then scaffold that all the way to 9 chromosomes of this crops or of this vegetable. So this is the synteny analysis, and of course, I think there is a few synteny or a few orientation that has to be checked. But again, it shows that with this genomes, with this chromosome level genome, it actually provide a more complete genomic information of these crops. Okay, so if you are interested to listen to this webinar by Dr. Richard Michel Moore, you can actually go to this YouTube site. It's a very, very comprehensive study with many versions of draft genome and how dovetail genomics help to improve and then get to the final version. Okay. Moving on to the second case study. So this one is the tropical fruit durian. Just a fun fact, durian is quite popular in Singapore and Malaysia, and of course in Southeast Asia. And we love durians. Okay, so the background of this project, durian is the king of fruit. It's grown in Thailand, Malaysia, Indonesia, and so far there's 13 known species of in this durian genus, and it's also one of the important fruits where China itself imports about 600 million worth of durians into China. Okay. And the objective of this project is basically to assess the phylogenetics relationships of durian with other Malvaceae members, as well as to compare the transcriptome data between a different plant organ. So basically, the groups look for a few organs, as well as the last objective is to use genomic and transcriptomic analysis to understand the evolutions and regulations of the fruit-related process such as fruit ripening, the flavor, as well as the sulfur metabolism. So again, only with the complete reference genome or complete genome, then you can perform downstream data analysis. So this species itself, initially there wasn't a reference genomes available. So basically this is building a reference genome. So to build a reference genome, the group used PacBio. So they sequence the plant to 153x coverage of the durian genome, and then we use Chicago and Hi-C to scaffold this draft assembly. So after scaffolding with Chicago and Hi-C, we were able to get to the assembly length is 750 megabases, which is quite close to the estimated genome size. And that is accounted for 96.88% of the genome. So anything that above 95%, that is good. So again, this shows that this genome is quite complete. And with that complete genome, the group would then look for other downstream analysis. So for example, they did the comparative phylogenomics to understand the relationship with other Malvaceae species, and also they also performed other experiments such as gene family analysis, pathway analysis, phylogeny expression, as well as synteny analysis. And again, with all this analysis, it provides available resources for biological research and agronomy of these tropical fruits. Okay, the last case study that I'd like to share with you is this cheatgrass. So this is a project that that's done with collaborations with BYU and WashU. So cheatgrass is it's not native to the US. It's actually imported a long time ago, and cheatgrass actually absorb water, and it makes the land very dry, and because of that, it's easily causing the bushfire when there is a dry season, and when it's a lightning, it causes bushfire. So the researcher or the group would like to understand the genome of this cheatgrass, as well as to understand probably how is it evolved when it's imported into the US. So again, to understand this species, they will need to get the genome or to obtain the genomic information. So again, to build the reference genome, as well as using dovetail technology to scaffold that. So the draft genome was done using PacBio. They sequenced that up to 87x coverage, and then they use Kernel, which is a software to assemble that, and then they polish that with and also using PacBio as well as Illumina reads, and after that, that the draft assembled was down to 68.6x coverage. And we dovetail use the Hi-C technology. We use two types of technology to scaffold. One is restriction enzyme based, which is dovetail Hi-C, as well as we use DNase based, which is OmniC. So we use two types of technology to scaffold this cheatgrass, and the result shows that we were able to scaffold that to chromosome level, and also with the comparison between restriction enzyme Hi-C and DNase Hi-C. DNase Hi-C is actually performing more superior than this restriction enzyme Hi-C. So at Dovetail, we have actually discontinued using restriction enzyme Hi-C, because we know that DNase Hi-C has much more superior performance. Okay, back to this one. These are the synteny analysis. So these are the two related species, and it shows they are quite concordant with each other. Whereas choosing another species that is unrelated, you can see that they are everywhere. Okay, and these are chromosome level assembly. So basically for this cheatgrass, there are 7 chromosomes being assembled. And for most of the chromosome, we were able to scaffold that from telomere to telomere. So you probably see this red line here at the end. So this red line represents the telomeric sequences. And you can see in this BT05, which is maybe chromosome number 5, we are able to scaffold that from telomere to telomere. Okay, so this is probably my last slide. As I mentioned earlier on, Dovetail Genomics is the leading company in this technology. We are like doing de novo assembly as well as the genome scaffolding, and there are many publications and many high impact publications that has been published using our technology. If you are interested, you can go to the website and take a look. And so far, we have more than 80 publications using the technology, and it ranges in many different species, from plants to animal to sea creatures to anything that maybe you can think of. So with that, I'd like to thank for your attention, and I'm happy to take any questions that you have. Please feel free to type in your chat. Thank you. Thanks for the attention.
Thanks a lot, Dr. Lee. I would request all the participants to drop in questions in the chat so that we can take them up, Dr. Lee. Dr. Lee, we have a question from Dr. M. Ganesan. He's asking, what is the error rate for longer reads?
Okay, so I believe you are asking for the, when you say longer read, is it PacBio and Oxford Nanopore? Or you are referring to the proximity ligation?
Dr. Ganesan, I hope he's talking about ligation. Yes, ma'am.
Yeah. Okay, great. This is a good question. So if you look into the technology, right, basically it is no different from Illumina technology. So when we talk about error rate, especially with OmniC, the error rate is actually similar to Illumina shotgun. So if you know Illumina shotgun has a very, very low error rate. The proximity ligation has the similar error rate to Illumina shotgun data. So we have the data show that our OmniC data is actually mimicking Illumina shotgun. So because we are actually taking these reads and then we are doing the Illumina library prep.
I think getting the PPTs would be a bit difficult, sir, or ma'am, whosoever has put forth this question. Meanwhile, the same session is being live streamed on YouTube, and you will have a recording for the same on YouTube as well. Whenever you want, you can refer to that. Link has been shared with you previously and would be shared again. So you can refer to the YouTube link. Okay, Mr. Rohit is asking, can we use dovetail with primary assembly made from nanopore with or without Illumina?
I'm not sure I understand the questions correctly. Dovetail technology work with any assembly data. If the assembly data is from Oxford Nanopore, definitely it works with Oxford Nanopore data set. However, as everyone knows, Oxford Nanopore data set has higher error rate, right? As compared to let's say PacBio. PacBio, they have this high five technology where the error rate is actually quite minimal, and the quality is actually quite good. So Oxford Nanopore definitely dovetail can work with any assembly data. And as you can also imagine that the input of dovetail is the draft assembly. The better the quality of the draft assembly, the better the outcome of the genome scaffolding. So if the Oxford Nanopore has high error rate, which has not been polished with Illumina reads, and that will jeopardize the downstream genome scaffolding. So garbage in, garbage out. So my advice is, if you are using Oxford Nanopore, and if you know that the error rate is high, my suggestion is to use Illumina read to do polishing, and then go with the downstream genome scaffolding.
Okay. In addition to the same question, Mr. Rohit is asking, what is the primary assembly completeness or N50 criteria for using with dovetail?
Okay, this is a good question. I think many researchers would like to get a hard matrix of this one. Unfortunately, it cannot be. I cannot give you a hard criteria of N50. Why? Because the input varies a lot. If the input is in kilo base pair, you can't expect that with dovetail technology that will get you to that will 100% every time will get you to chromosome level, right? So like what I said, the better the draft genome, the better the outcome of the genome scaffolding. So there is no criteria for N50 using dovetail technology. What I can say is definitely using dovetail technology, it will help you to scaffold, to put that draft assembly from maybe thousands and thousands of scaffolds, reduce that into maybe hundreds, or reduce that into lesser number of scaffold, as well as increase that contiguity, and also putting that order and orientation. That is the purpose of dovetail technology.
Okay, thank you for your explanation, ma'am. Ma'am, we have one more question coming in from YouTube. They're asking, how does this method improve over the currently existing method for structural variation detection?
Very, very good questions. Okay. So structural variant detection is another aspect of analysis. It's not in this genome scaffolding. It's not in this genome scaffolding workflow. But basically, after you have gotten the draft assembly, as well as you have gotten these types of information, let me take example of this one. So what you see here, what you see in this plot, I mentioned that each of these is basically one chromosome, right? Or we say pseudo chromosome, because you still have to do genetic mapping to ensure that this is the chromosome, right? But basically, most of the time, this is the chromosome level genome scaffolding. What you see in this plot, or outside of this plot, every single dot here, these are the Illumina reads from the proximity ligation. So every single dot is basically this dot. So this dot, when we scout map that to the draft assembly, and then we were able to join all the scaffold together and become this big scaffold, right? And for structural variant detections, after you have gotten this draft genome, let's say this draft genome is confirmed, is validated, and this is a good reference genome. And then using the technology, using the proximity ligation technology, if you run, let's say, different variety or different plants or different individual, you map that information back to the reference genome. So this is your reference genome. You map that back to reference genome, and if there is any structural variants, what you would detect is basically higher signal in a certain region, and it mapped to two different chromosomes. For example, if you see that there is a high read that map to this region, so for example this region as well as this region, so you know that this could be a structural variant of chromosome 2 and chromosome, let's say 1, 2, 3, 4, 5, 6, chromosome 6. So that is how you analyze structural variants. Yeah, but that is the theory, or that is the principle of using the technology in identifying structural variants. I hope that's clear.
Thank you so much for your explanation. We have one more question coming in from Mr. Rohit. He's asking how dovetail OmniC technology assembly stands when compared with 10x genomics based superquantic based assembly.
Okay, so as far as I know, 10x Genomic has discontinued their assembly product, that it is no longer available in the market to do this assembly assay. So it's no longer available. But compare that technology, basically 10x, we used 10x before, and 10x is to assemble the draft genome. It works like PacBio or Illumina or Oxford Nanopore. So we use 10x to assemble the draft genomes, and then using dovetail technology to scaffold the 10x genomics output. That is how we work using 10x genomic data. And if you think about the technology, if you think about 10x or PacBio or Oxford Nanopore or Illumina, the first step of this technology is basically to extract DNA, right? So you extract the DNA out, and then you will then use the DNA to do, let's say, Oxford Nanopore or 10x or PacBio. So you are sequencing that DNA fragment. But the proximity ligation technology, we work with chromatin. So we don't do any DNA extraction at the first step. Immediately, we cross-link the chromatin. So the information that we are getting is basically chromatin level or chromosome level. So the length of the information that we are getting is actually longer than the previous technology that I mentioned, right? Be it second generation or third generation technology. So that is I would say probably the difference. So our technology is to scaffold. We work with 10x data before. We work with Oxford Nanopore. We work with PacBio data as well as Illumina data. So all these data are draft genome. Yeah, it's not. So I would say we complement this technology. So we complement, be it Illumina, PacBio, 10x Genomics, or even Oxford Nanopore. We complement this technology, and then we scaffold that draft genome that's coming out from that technology.
Thank you so much for the answer, ma'am. Any more questions, guys? You can please feel free to type them in the chat section. Any more questions, please? We'll be more than happy to take them. Yeah, another question, ma'am, we have one more question from Dr. Padaru. I think they're asking, can you please explain more about the software analysis part involved in this? I mean, how different would it be from the regular approach that we take for any genome assembly analysis?
Okay, so I have this slide, which is a very simple explanation. The analysis is, so this slide, I hope, will explain the fundamental or the principle of the analysis. So basically, the genome scaffolding is the second step in the building the reference genome, right? So the first step of building the reference genome is to get the reference genome. So you get the reference genome here. After you get the reference genome, so you sequence, you get the reads, you assemble that using software such as Canoe or SOAP de novo, or there are many software out there that you can assemble this read. So you assemble that read, it forms scaffold, right? And with dovetail technology, now we take this draft assembly, which is a scaffold, as the input, right? And then we combine that with the proximity ligation reads. So these proximity ligation reads are generated from this workflow. So here, after we do this proximity ligation technology, we take this DNA and then we prep that using Illumina protocol. So now the library is Illumina library, but it's not typical like short reads because there is this proximity ligation happen upstream here. So when we do these library preps, the reads are actually very far apart. So these are the reads that has the proximity ligation happen, right? So we take this proximity ligation read and map that to the draft genome. So just imagine that this is a draft genome, right? This is scaffold number 1, scaffold number 2, scaffold number 3. And these are the proximity ligation reads. So we sequence 2 by 150 base pair. So these reads, when I map to this whole bunch of draft genome, these reads may be mapped to these sequences here, read 1, and then read 2 mapped to the sequences here, right? Okay, so there is a connection here, but this only one read doesn't show any, it doesn't have any meaning, right? So I have many reads, like what I mentioned, there are millions of reads, millions of proximity ligation reads. And with the same principle where when I map the reads, it mapped to read 1 here, read 2 here. This one mapped to read 1, mapped to read 2. So now I have, for example, 3 reads that telling me that these 2 scaffold, they are coming from one single strand or one chromatin, right? So with that principle, with the million reads that joining the scaffold together, now I'm able to put all these scaffold into a bigger scaffold. So for example, I'm able to put this few scaffold into chromosome number 1, this few scaffold into chromosome number 2, and that resulted in this one. So these one single plots are actually joined from many scaffolds, and with these joints here, now we know that they form one big scaffold, and there is no interaction of this scaffold to this scaffold. So we know that this is one chromosome, this is another chromosome. So I hope that explained the fundamental or the principle of the data analysis of genome scaffolding. Of course, the software itself can be, there are many steps in the software itself. To run this, first of all, you need to have the 2 inputs, and then there is a mapping, there is a corrections, there is also the model generations. Once the model generation is done, then there is also many rounds of this scaffolding occur until the software like can't find any join, and then there will be break as well. There will be break and join that happen in this whole analysis. Step. It's more complicated than what I'm showing you here, but in terms of the principle of genome scaffolding, this is basically how proximity ligation reads help to join that scaffold.
Thank you for your answer, ma'am. I think Dr. Badru, your query would have been satisfied by this answer. Ma'am, we have a question. I'll just divide it into 2 parts. It's coming from Ms. Monica. Her first question is, how much tissue is required for this particular approach, in this particular approach for library preparation? That is her first question. The second question is, what challenges are faced during the sample processing steps? I mean, till library preparation, whatever sample preparation protocols that are involved, are there any typical things that we need to keep in mind while carrying about with that process?
Okay. Okay, so in terms of the material required for this approach, it's not a lot. If I remember correctly, it's one assay probably require 150 milligram of the plant, of the leaf, young leaf. Again, if you can check on the protocol, if you are interested, we have a very, comprehensive protocol available. But I remember it only require maybe 150 milligram to maybe 300 milligram of the plant, of the leaf. So that is the sample requirement. In terms of what are the important steps to take note of, right? This assay is quite sensitive to DNA degradation. So when you harvest the leaf, ensure that you harvest that and immediately freeze in liquid nitrogen, because if you harvest and leave it at, let's say, room temperature, or leave it at maybe 4 degrees, the chromatin will be denatured. The chromatin will it will deform, denature. You will not get these types of information. So preserving that chromatin contact intactness is very, very important. So so that is one important point to take note of. Is the harvesting. You harvest the leaf, immediately freeze that in liquid nitrogen, and do not thaw it until you want to start the experiment. And also plant. There are many types of leaf in plant. There are leaves that is quite thick. There are leaves that contains a lot of phenolic compounds, as well as there are plant that quite jelly type. For example, aloe vera. Aloe vera can be quite jelly. So these are all the important points to take note of. What types of plant species that you are working with, and if you are interested, you can talk to Bionivid and see they can give you some technical consultations, or you can write to us as well. So again, these are some of the important points to take note of. Which species are you working on? Because different leaf will have different compounds, or maybe substances that might be you need more care in terms of processing the sample. Other than that, if this is normal leaf without too much of phenolic compound, it is actually quite easy to get the assay done, because it's like molecular biology techniques. So you cross-link with formaldehyde, you digest with enzyme, you end repair with the T4 DNA ligase, and then you this ligation step is basically using ligase. You reverse cross-link and then library prep. So it's molecular biology techniques.
Thank you for your response, ma'am. We have another question. I'll again split it into 2 parts. The first one is, are there any specific suggestions or attention required for handling polyploidy, like tetraploidy in plant genomes? And secondly, does this particular technique help in assembling or segregating parental alleles? That are the questions that we have got.
Okay.
Yeah.
So I think this is very common questions asking about assembly of polyploidy. As you can see here, the data into genome scaffolding is basically the outputs of draft assembly. If you are able to separate the polyploidy here at the draft genome, you separate that draft assembly into different polyploidy, then use that as an input into dovetail, we can scaffold individual polyploidy. Okay, so that is one way to do it. You separate that polyploidy at the draft genome. There is also this paper that you might probably would like to refer. This is the strawberry with the octoploid, and this group actually using our technology to scaffold this octoploid strawberry. So I would suggest that if you are interested in polyploidy genome scaffolding, you can refer to this publication that published in Nature Genetics. Okay, and also with the OmniC technology plus PacBio Hi5 reads, which I'm not presenting here, but we have demonstrated that using PacBio Hi5 reads plus Dovetail OmniC technology, we are able to do the diploid assembly in human as well as in tuna. So when I say diploid assembly means, okay, let me give you more background. We know human are diploid, right? Father and mother. As well as tuna is also a diploid with paternal and maternal haplotype. So we use these two sample, and then we try to scaffold that into haplotype assembly or diploid assembly. So we using PacBio Hi5 reads plus the OmniC, we even without the paternal and maternal genetics information, we were able to separate that assembly into two haplotypes, into the maternal haplotype and paternal haplotype. We demonstrated that on tuna, and this tuna sample was actually a wild sample that catch from the sea. We were still able to scaffold that into two haplotypes, and so far we only demonstrated that in diploid genome. We have not done these types of project in polyploidy, because polyploidy can be quite challenging. But with diploid, using PacBio Hi5 and OmniC, we were able to scaffold at least the data that we work with. We scaffold that in human as well as tuna. Okay, so that will be my suggestions of polyploidy assembly. The second question is whether are we able to separate the allele, right?
Yes, the parental alleles, yes.
Mm-hmm. Yep. So I think to be honest, I'm not very sure. I'm not very into the downstream data analysis. But I believe that with the OmniC, because OmniC mimicking Illumina shotguns, where it actually helps it can actually use to do phasing. So after getting the draft assembly, after getting this whole complete genome, and then use the OmniC data to run phasing, I believe you are able to separate that into parental allelic information. We know that with OmniC data sets, we can do phasing analysis to separate that into maternal and paternal haplotypes. So I think you can use OmniC to do phase analysis to separate that allele information.
Okay, I think that must have definitely answered the said question. I think we don't have any more questions left with us. Thank you so much, Dr. Lee, for taking this time out and being a part of this webinar and addressing the entire participants and enriching our knowledge about this path-breaking technique in the field of genome sequencing. Thank you so much, ma'am.
You're welcome. And thanks everyone for your attention. Thank you.
So this brings us to the end of today's session. It started with welcome notes and presidential addresses from Dr. Jayasekar, Secretary ISPC, Dr. Maheshwarappa, President ISPC, and Dr. Anita, Director CPCRI. Then for the very first talk, we had Dr. S.V. Ramesh throwing some light on how genotype specific transcriptome, responses can be used to enrich the entire genome resources which are available for coconut research, right? Then we had Dr. Awadh Shah talking about how the GBS sequencing approach and how it is applied in a wide range of plant breeding programs like Jiva studies, linkage analysis, etc. Then we had Dr. Lee from Dovetail Genomics talking about how the various offerings from Dovetail Genomics, in particular the OmniC technology, can help in improving genome assemblies and resolving critical structural variations. This was what she discussed, and all of them handled a lot of questions coming in from the participants. So we would like to thank all the speakers for taking out this time from their busy schedules and being a part of this webinar. We'd also like to thank all the participants. At peak, we had around 35 participants joining in. We'd like to thank them for sitting patiently through the session and putting forth all the queries that were quite relevant to the theme of this webinar. We would be starting at around 10 a.m. tomorrow. So I would request all of you to be present tomorrow as well. The same link which was used today would be used for tomorrow's session also. You would be getting a mail from us today as well, which will bear the YouTube streaming and the webinar links as well. So kindly be there. We have also added the feedback form for today's session in the chat. I'd request all of you to just go through it and provide us with your valuable feedback. That is pretty much it for today's session. Thank you guys. Thank you once again. Thank you to all the speakers. Thank you to all the participants. Thank you to ISPC members. Thank you for making this day grand success. See you tomorrow, guys. Thank you so much. I've shared the feedback form again in the chat. You can please have a look at it. Also, the same would be shared in the various WhatsApp groups that we have created. You'll be getting the feedback link there as well. Right? Thank you so much, everyone. Thank you for your time. See you tomorrow 10 a.m.